Assalamualaikum and hi to Dr. Wan. Today is this is my last video for this semester for Microbiology 1. Dr. Wan asked us to make video and summarize all topics that we have learned for whole semester. So I would like to summarize about microscopy. Microscopy has two categories of microscope, which is light and electron microscope. Light microscope has 5 types, which are bright, dark, contrast, fluorescent, and confocal. And then electron has 2 types, microscope, and I would like to summarize about introduction to microbial world. We learned about the first microbiologist, Leeuwenhoek. He was the first to observe cells, animalcules, and Edward Jenner, who Needham, and also postulates that explain about a disease pathogen that cause disease must be the same, and then we learned about introduction to prokaryotes, which can categorize to divided into 2, archaea and bacteria. We learned about cell wall that have peptidoglycan. Most of bacteria have peptidoglycan. Bacteria gram-negative and gram-positive bacteria. Staining bacteria, we identified them by gram staining, which we learned in microscopy chapter. And we learned about intracellular structure of prokaryotes. Dr. Wan also asked us to make a mind map about this topic. Intracellular prokaryotes is all about membrane-bound organelle that bound in the cells, like plasma membranes, ribosomes, Golgi apparatus, and then we learned about classification of organisms, the taxonomy. Taxonomy have 3 parts: nomenclature, identification, and classification. And we also learned about virus. We also make a mind map about this chapter too. Viruses are cellular agents. It needs host. It has envelope and non-envelope virus, and then we learned about genetics that have frame shift mutation, and then have nonsense mutation, stop codon, UAA, UAG, and UGA. And then we learned about nutritional types of microorganisms that have two major nutrition concerns, source of energy and source of carbon. And then we learn about the proteins. Proteins have three groups under proteins: protozoa, algae, and slime molds. Algae have can divided to rhizopoda, Eubinophyta, and more. And then we learn about epidemiology. John Snow was the first epidemiologist. Epidemiology is all about how to control of health and disease in defined human population. And then we learn about fungi. Fungi that have a rigid cell wall, chitin. Fungi also have a hyphae, mycorrhizae, and then algae also fungi also combine with algae, and fungi also combine with plant. Fungi that combine with plant we call mycorrhizae. And then we learned about genetics, recombinant, they have enzymes, ligase, lyase, oxidoreductase, and some more. And then we learn about microbial metabolisms. Microbial metabolisms is all about phosphorylations and photosynthesis, and aerobic respiration, and anaerobic needs carbon dioxide, while aerobic respirations do not need carbon dioxide, and then fermentation for cells that cannot respire, cannot do respiration. And then we learn about control of microbial growth, sterilization and disinfection. Sterilization that kills, removal of all microorganisms. Biocide and bacteriostasis difference because of biocides kills microbes, while bacteriostasis not killing the microbes. And then we learn about antimicrobial chemotherapy that have chemotherapeutic agents that are most antibiotic. Most of them are antibiotics. And then we learn about microbial growth. For last chapter we learned about microbial growth. And then microbial growth, we have anaerobic incubations, subculture, and then we learned about lamina. And then we learn about agar. We learn how to make medium and nutritions and all. So I think that what I understand about microbial growth. I cannot make this video more longer because of time also. Time that Dr. Wan gave a short, and then I want to thank Dr. Wan that taught us for this semester, and I hope that we can learn more, we can gain more knowledge from her in another time. So thank you, Doctor, and wish us, not in our final. Thank you.